All right, well, in interest of time and getting to the main event here tonight, I'm going to go ahead and get started. For those of you joining us and about to join us, I'd like to welcome everyone to the fourth installment of our Bio5 Precision Wellness in the Time of COVID event series. My name is Lisa Romero, and I'm the Senior Director of Public Affairs, Communications, and Engagement for the Bio5 Institute. Thank you all for being here this evening. Bio5's mission is to connect and mobilize top researchers to find creative solutions to humanity's most pressing health and environmental challenges. Since 2001, this interdisciplinary approach has been an international model of how to conduct collaborative research and has resulted in disease prevention strategies, promising new therapies, innovative diagnostics and devices, and improved food sustainability. And it includes the precision nutrition and wellness initiatives as one of its focus areas. Tonight's event and the larger series is part of that wellness and precision nutrition and wellness initiative. And the mission of precision wellness, I can't speak tonight, I'm sorry, is to determine and optimize the state of health of individuals in order to predict and prevent disease and increase overall quality of life. This is achieved through integration of scientific research, next generation technologies, and personalized nutrition and lifestyle design. We also value the power of education in promoting health. So in this series, we've chosen speakers that are at the top of their scientific game, but who are also relatable to all of us joining the conversation tonight. So we have a great talk for you this evening presented by Dr. Yves Lussier, who is the chair of biomedical informatics at the University of Utah School of Medicine. Dr. Lussier is a result-oriented expert in biomedical informatics whose research integrates health information, technology, and bioinformatics to individualized disease treatment. Dr. Lussier is going to discuss the role of single-subject genomic studies in precision medicine and wellness. So audience members, please go ahead and type your questions throughout the talk in the Q&A box located in the bottom toolbar on your screens. We'll be sure to leave time at the end for your questions at the end of the presentation. And with that, let's get to the good stuff. Dr. Lussier, we're delighted to have you, and please take it away.
Thank you. It's a pleasure to be with all of you tonight. Let me just start this presentation. Currently, there you go. Lisa, is everything all right? Can you see the slide properly? Hello?
Yeah, all good, all good.
Okay, very good, very good. And in the context of precision wellness, so my expertise is in medical care and computational medicine. So we'll be focusing on these computational approaches to precision medicine. Particularly, we'll drill down into the advantages of single subject studies. There's a few former conflict of interest, but right now I'm not on board of these for-profit companies. I've got many trainees in different institutions. And I'd like to thank the University of Arizona, with whom I've had the opportunity to conduct research at Bio5, and also to participate in the Precision Medicine Initiative, which is a way to get your own genome paid for by the NIH. You don't need to go to 23andMe. You need to give a bit of blood and your clinical data, and there are a few other agencies that also support me. So first, let's try to imagine if you could investigate and mine very large data set and discover through practice-based evidence rather than prospective research, retrospectively, some unexpected side effect of a treatment or comorbidity progression of COVID, for example. I'll be showing you evidence that this can be done with large data sets that are available right now. And in addition, how can you generate at the opposite when you have very few cases and rare and infrequent conditions, what are the other approaches at the other end of the spectrum? Instead of having very large data set, having very few data, just on a few patients. And I'll try to show you how computational approaches can address these two apparently diametric extreme conditions. And rapidly, a preview of what I hope you'll learn from this presentation. There are approaches that can lead to biomarkers and identifying, for example, genetic testing in Mendelian diseases has been very powerful. A single gene is responsible for a disease of genetics. In more complex inheritance, and especially the response to COVID, this is often at the pathway. The problem is at the pathway level. And I'll be showing you how we can integrate signal at the pathway. And that's called the blessing of dimensionality, when you have a lot of measures in one person and you can actually discover signal there and give significance to this signal. Additionally, the approaches to do that, you sometimes want to challenge tissue from this person and see the response. For example, the response to COVID ex vivo. We could take the blood, and how can we derive a response that would be unique to one person and show that it's different than others from an ex vivo assay that could be predictive, perhaps, of a severe complication, for example, to COVID. We'll be using other viruses to show you that, but this will be what the presentation will show. So first, let's address how can we take large population data sets and even biomarker data sets and mine them together. And as you can see here are chromosomes that fall onto Earth, and we see a few people over the continents around down there. The data set I'll be using here will be the UK Biobank. That's a biobank offered from the United Kingdom, and they have a subset with COVID data. There were about 500,000 subjects in it with availability of COVID results. 67,000 had cancer, and of these, a smaller subset were hospitalized with cancer. And the paper was published recently, just a few weeks ago, in the British Medical Journal of Healthcare and Informatics. And we're looking into the impact of COVID-19 for patients with cancer, the survival, and how it changes if someone has both COVID and cancer versus cancer alone. Oops. And these are the results. Rapidly, you have the cancer names to the left. Each cancer was studied separately, and you have the number of cancer cases. When there's very few cases, the results have large confidence interval to the right. If you see to the right, the panel there, if it crosses the red line, that means it's not significant, but if it doesn't, it means it's significant. The median or the average response of the effect is the square in blue, but you can see sometimes very large confidence interval. For example, the first line, localized cancer with 3,600 cases, 235 of which having COVID, we saw that the fatalities among those that had COVID-19 were 19, while there were 2 among localized cancer in the absence of COVID. So obviously there's a high, as you can see, there's a high risk of cancer. The hazard ratio is over 8-fold. So it's a much higher risk if you have COVID and a localized cancer to die during a hospitalization. And we can see for other cancers where they belong, starting with those that had higher risk, like lymphoma, melanoma, and leukemia, and those that had the smallest risk at the end, bladder cancer. There doesn't seem to be a risk that's different from no changes, which would be a 1. A risk of 1 means there's no increase or reduced risk. What I'd like to point out, though, is if in addition you have metastases, what you see at the bottom to the right here, these numbers are additive to those to the top. So if you, in addition, you have melanoma, let's say, to start with, you're having a 16-fold increase of risk of dying with COVID-19 and melanoma if hospitalized. If in addition you had metastases, you have an additional risk of 32 that adds to that. So these are additive risks to the bottom here to those of the top. Again, it's pretty impressive how metastases can add severity, but metastases are the cancer that progresses in a distal organ, not the organ of origin. This is a second paper that illustrates going to the molecular level. And this one here, we're trying, and it was published in September, we're trying to show how we could explain molecularly why asthmatic patients had an increased risk of dying from COVID. And the idea was pretty simple. As you remember, COVID focuses its entry point with a receptor called the ACE2 receptor of the cells. And what we've noticed is that subjects that have severe asthma versus those that have a moderate or low asthma are more likely to have an increase in the receptor presence, and particularly if they're co-infected with another virus, such as the rhinovirus. And that's what the data to the right is showing here. And the data to the left is verifying which of the altered expression of the downstream products of the genes. So the genes, as you remember, or the DNA is the blueprint of the human being, and that's relatively static, except in cancer. But in this case, for asthmatic people, that's static. What's dynamic is that it codes for messenger RNAs and protein. This study here is done on messenger RNAs, which are the vehicles by which the machinery of the cell decodes and makes proteins. It's also the area where the virus interferes with the cell in order to do its own proteins. And what we're looking at are all the expression of the messenger RNAs, or the downstream products of the genes that were correlated with ACE2 in those severe asthmatic patients where they were different from the non-severe ones, and which kind of downstream cytokine production occurred. And we could see that for the severe asthmatic patients, a lot of interleukin, which made an interferon and TNF alpha and TNF production were actually mediated and increased. And these increase the cytokine production, which is the response of the organism to the virus. And as you probably remember, there are different ways of dying of COVID. And it was in the press early on. The physicians were having a hard time in Mars figuring out of the epidemic, not this year, last year, figuring out how to treat these patients. But we figured out if we can understand if they are actually overreactive to the virus, we should give them steroids and reduce their immune response. And if we take them early enough, we can then give them antivirals. But the antivirals are not sufficient to stop the disease if it's the reaction of the body. So that requires addressing the cytokine progression. Our studies were going to much more sophisticated network. We were going also to the molecules themselves. But you can see now, as you look into 29 genes and the mechanisms that are affecting these asthmatic patients in response to rhinovirus, and how that interacts with as a comorbidity with COVID. And there were reports that were showing that people that had the common cold, like rhinovirus, were dying more of COVID than this if they were not co-infected. These reports came from Stanford, and that's in a response to trying to understand what's the mechanism of these comorbidities that kill the patients faster. And but again, this is generic over 30 patients. How can we get to a single patient? So this is a figure that appeared 20 years ago when the first human genome that cost $1 billion to calculate was first published. And there was this idea that you could capture with a net a large number of genes, perhaps working in unison into a pathway. So there are many types of pathways in the body. There's metabolisms that have pathways. There's also signaling from the cell receptor to the nucleus, which is where the virus is working, and whether these pathways would not be more useful as biomarkers than the conventional method, as the fisherman to the left, that's just going with one gene at a time. There's evidence that perhaps we should change the model. There's been 30,000 single molecule biomarker grants in the last 25 years that were funded by the NIH, over $2.5 billion. And we know it's difficult to find all the data, but between 2012 and 2017, there were only 12 new FDA-approved single molecule biomarkers for cancer. So now the question is really, should we change the paradigm and look, instead of a single transcriptome that would be concordantly altered across patients, look into patient-specific biomarkers where a pathway could be altered, but in a different way between, let's say, a certain patients and another one, yet it's the same pathway that's affected. That means that we relax the condition. It's not a single molecule that's always the cause, like in genetic diseases, like Mendelian disease, like cystic fibrosis, but instead it could be different molecules in different people, but the summative effect of these molecules would add up together to give this pathway effect. And there's other evidence for that. The first Mendelian diseases that were understood were in the mid-1800s. Hemophilia. The royal family of Great Britain suffered from prolonged bleeding time, and they wanted to understand what it was. They paid the first genetic research in Great Britain, and it was on hemophilia. And everywhere in the pathway here where you see a circle, a mutation of that gene that's called a loss of function, that gene is not function, the corresponding protein would not function, leads to downstream to a prolonged bleeding time, because a polymer that's supposed to, a fibrin that's supposed to create a clot and stop the bleeding is not forming as fast. And it could have happened in both sides, the extrinsic pathway or the intrinsic pathway. If the both are affected, it's actually lethal. No humans can live with both parts affected. In these cases, these are all Mendelian diseases, which are single gene disease. It's a little like a crystal where a diamond, if it's a bit yellow, people would know exactly which other atoms but carbon are involved, or if it's blue, or there are other atoms that create that color. And we can see that macroscopically at the naked eye, while the problem is molecular. In the same way, a little like a diamond, Mendelian disease reveal themselves at the organism level, but are caused by a single cause downstream. But what I'll show you is that there's another known condition that has prolonged bleeding time, and it's a chronic fibrosis of the liver, often caused by alcoholism. And in this case, there's absolutely no mutation of any of the genes. The proteins are totally normal, but there's less expression of these proteins in the liver. And because of that, the coagulation is prolonged. In this case, it's the summative effect of 3 proteins that are less expressed in absence of any Mendelian mutation that gives exactly the same intermediate phenotype that's common between hemophiliacs and people with chronic liver disease. Again, the prolonged bleeding time is a pathway disease. It's not a single gene disease. That's the point I was coming to. And it's not necessarily always the same molecules. In this case here, we're speaking of the expression that's altered, while the other one was a single point mutation, which meant that the protein could still be produced but dysfunctional, or it's a knockout, the proteins that produce at all. So this brings me to another discussion. How can we get to single subject studies? Conventional studies require a lot of patience because we're trying to find the average effect. And as I pointed out, the biomarkers have to be measured and be concordant at the molecule level between patients. If we relax it and we don't require the molecule to be concordant, but the pathway to be concordant, and we don't require to look at that across patients, but we serve as our own control, we believe it's a better strategy. While for the static genome, the DNA, I think the next slide will explain that, if I can get to the next slide. Well, we can accumulate these data at the pathway level, and there are different databases that are either biased or unbiased where that can be done. This is a bit of jargon, but it could be at a different number of type of mechanisms in biomolecules of life. But this is the slide I was trying to get to. If we think of the top here, the DNA, that's a blueprint. Except for cancer, the DNA is generally static. And having a reference DNA from the genome of many people allows us to compare a single person, so single subject analysis here on the top, to that full genome and identify what are the variants. So there's a possibility to do a single subject study of the DNA that's quite standard, and the geneticists that have identified new diseases have often proceeded with this strategy, and we're lucky because the chair of this session, Michael, is one of such a discoverer of new diseases with these methods. Not that it's easy. This remains very difficult, but at least there's a process. For the dynamic part of the genome, it's even more complicated because a reference transcriptome, which is the RNA, or a reference metabolome would not really work. Every time you change the template, the blueprint on the top, it gives a new normal. Every one of us, to the naked eye, are different, right? Anyone in the street would know that. And we could be totally normal and disease-free and be different. At the cellular level, the same thing occurs. There's a slightly different blueprint in each of our cells than that of someone else besides you. In addition to that, cells are specialized. Otherwise, we'd be a big blob like a sponge of a single type of cells, but we're not. So as they specialize, they don't express everything that's in the blueprint. The blueprint has what it takes to make cells and bones. It has what it takes to make the skin or a nerve, and these cells are quite different. So comparing a tissue to its normal, to its comparison in the same individual, we believe it is the best way to produce and show single subject studies. But this brings really a problem. How do you get to large cohorts from single subject studies? And I'll show you how machine learning can be applied to single subject studies to the right here, or even meta-analyses that were developed in the past to compare different studies together of multiple patients. It's actually applicable as well to single subject studies. And this gives the advantage that studies which in the past would have required 150 people to do a machine learning classifier require only 20 people. And I'll show you a prospective study where we did that with Dr. Fernando Martinez of Bio5, former director of Bio5 actually. And we'll show you also how that can be done to compare two groups of very small groups of patients, which traditionally would have required 20 or 30 subjects per group. And we can achieve that with perhaps 3 to 4 subjects per group. So this is the approach. The approach is that we take a healthy tissue or tumor tissue of one person, or a healthy tissue, and we subject it to an assay in a Petri dish. And then we look at each gene, how it changes. A fold change is the expression of a gene product, protein, metabolite, or messenger RNA, divided by its value in 2 different conditions. And then what we expect is that there would be some that are unaltered, shown here to the left. A fold change of 1 means that it's the same value in both conditions. And the log of a fold change of 1 is 0. So this is a distribution near 0 here. And otherwise, if it's altered, it's much bigger. And if we look at these two and we compare them in a pathway, you could find what's called an overrepresentation statistic. Is there more than expected? The genes in the pathway that are altered are more numerous than expected by chance or by a proper control. These type of statistics lead to the following discoveries. This here is a wheel that represents different pathways. For example, chromosomal localization here, homeostasis, DNA and chromatin assembly, and hormone secretion. And out of a study of about 55 lung cancer patients, there were 5 that were disease-free after 5 years and 5 that died of disease within 1 year. Everybody else was somewhere in the middle. And the color, when it goes to the periphery of the wheel, it means that the pathway is 10,000 times overexpressed. And when you see no green and it's a gray zone, it means it's abrogated. There's no expression of the pathway. And you can pretty much see that those that died of disease in less than 1 year had overexpression of DNA and chromatin assembly and chromosomal localization and abrogation of homeostasis and hormone secretion. While those that were disease-free at 5 years, the gray line here is where the pathway is unchanged. And the majority had nearly no changes in these pathological pathways that lead to death by disease in less than 1 year. And that is showing the power of these studies, because in each patient, you can actually read the output and see what's going on. It's not an average between patient here. You see what's going on per patient. You can even sum it as a histogram here, each patient being a row, and looking into these lung cancer patients, which ones had altered pathways. When you have less than 20 patients among the 55, all the gray region here, these pathways are invisible to traditional methods of investigation. It would, among these 55 patients, it would take a cohort that's much bigger to see these pathways. With 55 patients, it would take at least 15 to 20, like the purple regions, the blue, and the green. What does it inform us? If you look at immune response here, there's a lot of patients right here, that's a dot, the black dots, that have there's less than 5 patients in very different methods by which their immune system is affected. And this was traditionally invisible to the methods. What could be found is this region here of the immune response, where there's about 20 patients that are altered. So you can see, if we think of an iceberg where conventional methods would show you this part, there's a much bigger part under the water that's invisible by conventional methods of analyses. This is how we can compare. The conventional methods are in gray here, and 0 and 0, that's a very poor accuracy. You're trying to be to the top right here for the maximum accuracy. 50% recall, it means what you should have discovered, half of it was discovered. And at 90% precision, it means you're 90% correct in what you're testing. That's a pretty good statistic here in 5 versus 5. And that's a method of a meta-analysis that we call inter-N of 1 between the single subject studies. So it's comparing 5 subjects to 5 others. The lines that you can see are the dispersion when we randomize these patients among a large cohort, because each patient will contribute a bit differently. So there's a variation there. And you can see by conventional studies, the gray ones, when it touches near 0 here, it's useless. So it takes 9 patients here. So we can reduce nearly by half. At 4, you start getting a signal for these studies. This is the prospective study I was speaking of, and we'll complete the presentation with that. Conventional transcriptome level, messenger RNA level, machine learning algorithms require 150, 200 patients per group, and you usually need a prediction group, and you need to confirm it to show it's scalable to other patients with a discovery group that's totally independent. In this case here, we're not at 100 or 200 patients per group. We've got 10 per group. We're learning the response of, we're trying to predict which pediatric asthmatic patients will be hospitalized in the coming year among 21 pediatric asthmatic patients that are all very severe at maximum treatment. They were clinically indifferentiable. I'm not showing all the slides of their age and gender and weight, but when you look at which ones are hospitalized and which ones were not, they were indifferentiable. There's even tests for the lung function. They were all severe, indifferentiable for clinicians. At the molecular level, at the beginning of the year, you just need to take their blood, expose it to the common cold, which in pediatric patients is known to cause 70% of the hospitalization in severe patients. And you can actually measure their pathways that are altered in each patient, and subject them to the classifier that was learned on another cohort on the top, and try to determine which ones were exacerbated, were hospitalized, and which ones were not. And here are the same wheels I was showing you before. And each wheel represents one patient. That's also a contribution we've done in this paper 3 years ago in what's called interpretable machine learning or explainable AI. The majority of these machine learning algorithms are black boxes where you can't understand what's going on. And we've used one such system called random forest. But with our methods, since we get a p-value, which is the significance, and an effect size, which is how much it affects someone, for each pathway before subjecting it to the classifier, we can peek under the cover of the classifier and look at each patient and determine, according to what's called the features by which the classifier classifies. When you color these features here, and you can see in orange here, acquired immunity, it's orange here, innate immune response in purple is here, and so on and so forth, you can see what's going on. And those that we appropriately said would be hospitalized, these 9-1, and we're, according to the clinical phenotype, indeed hospitalized, where we were true. Look how they look like one another here as compared to the diametric extreme one that were not hospitalized and were predicted correctly. We also know why we failed to predict these three and these three here. This was a very small cohort. Would have performed much better would we have had a larger cohort. And also we designed the classifier over a related condition, but not asthma. We designed it, if I return one slide, over a Duke sample that was available, where they infected their students with rhinovirus, took their blood before the infection, 2 days after, and they observed that they were all shedding the virus in their secretion, but only half of them had symptoms like headache and runny nose. The other half were totally asymptomatic. Now that comes to no surprise to you that because we're in a pandemic like COVID, that half people that are infected could be asymptomatic. And that's what we've learned here. We've learned the mechanisms by which someone is symptomatic, overreactive, or less reactive, and it was predictive of the overreactive one in asthma, and that was our hypothesis coming in. Why did we not take asthmatic patients? It costs hundreds of thousands of dollars, millions to follow these cohorts, and my colleague Fernando Martinez only had 21. So in order to be honest, we had to do it with an independent data set and test it only once, and it worked the first time we tried it. So coming back to the preview of the take-home messages, the idea again was to show you how computational medicine methods could look into very large data sets and find patterns. We've shown you how the UK Biobank and COVID were predictive of increased hazard ratios for cancer patients with metastases and with certain type of cancers such as melanoma, lymphomas, and leukemias. And while it was not affecting patients, for example, that had prostate cancer. And we also have shown you how molecular pathways were important in determining certain diseases that are maybe not at the single molecule level, but maybe a pathway problems with, for example, a cirrhosis of the liver. And then we probably leverage this idea to measure that in high throughput in single patients, which then can be brought together in cohorts that are much more significant because we get a lot of power in each single patient as compared to conventional methods. What it does require is a lot of measurement, which we call the blessing of dimensionality. We spoke of 2 measures in each patient. Conventionally, there's usually just 1 measure of the transcriptome of the genome per patient, with very few exceptions. Designs are looking into that. So we need to change the way we investigate in order to leverage that. It's also scalable to the metabolome and the proteome and other domains than just the genome. And it's really a classifier of genome by environment as an interactions, because really we're looking into how the genes respond to either the stimulus or a different environment and how they're different in 2 conditions for the same patient. Bottom line is that we could really democratize the cost of precision medicine. Currently, the cost of measuring a whole genome or a whole transcriptome are reducing, but they still require very expensive machinery that's just not available in the third world. But every hospital in the third world has technicians that know how to do a technique called qPCR overnight. It's very affordable. So if we can reduce a classifier to the 20 molecules of life that are important to be measured overnight by qPCR in two conditions, someone could use a simple cell phone, plug in the data that they've measured by qPCR and get the answer from their own cell phone as to whether someone is classified as at risk of having, let's say, a severe COVID disease or not. And this is the opportunity for the future. And I remind you all that the first patient treated with insulin, I'm formerly from Canada, was in Toronto in 1920, and it cost $1 million in 1920 to treat that patient with insulin. And nowadays, insulin costs less than 10 cents a day for treatment of diabetic. So clearly, it may cost a lot early on, and the first human genome 20 years ago cost us $1 billion, and nowadays having a genome costs maybe $1,000. So we're already 1,000 times cheaper. It's the democratization of cost of precision medicine is going in an accelerated scale faster than the reduction of cost of computers. And on that, I'll be waiting for your question. Again, I'll just thank all the team that contributed to this and my collaborators from Bio5, such as Walt P. Gorsh, Helen Zhang, Ed Bedrick, Fernando Martinez, and John Galgiani. Thank you.
Thank you very much, Yves. My name is Michael Hammer. I'm also an associate director at Bio5, and I will be taking your questions, maybe posing a few myself. We do have a question from an attendee. I will try and interpret it with, I'll read it with one interpretation. It says, if you were provided with a specimen of skin showing malignant melanoma with a clean margin of normal cells, so I assume it's meaning both the melanomal cells and the margin, could you enhance understanding of the potential for metastasis?
Actually, I didn't give you enough detail on the long ones, but that's what we were doing there. In the long data set, we're looking into not normal cells, but the margin cells. All cells of the body, when we have a cancer, may be affected by the cancer. So we call that control is generally called unaffected cells. They're not affected by the cancer, but they may be affected by gene products of the cancer. So we're looking at the margins of that cancer versus the cancer. And these were the results we're showing, and we're able to predict, and that was the first study of single subject studies in using the N of 1 pathway, this idea of two measurement in 2013. It also won the best paper of the international conference that time. We're pretty proud of that. It was groundbreaking 8 years ago. And it was exactly a proof of concept using that and in many different patients. And you could see the patterns, if I go back there, of the patterns. Oops, if I rapidly go back, this pattern here on the top, a disease-free after 5 years or death of disease in less than 1 year. This is lung cancer, and though you could die of the primary lung cancer if the mass occludes both lungs, I've never seen that. Generally, you die of complications of lung cancer, the metastases. Generally, the cancer would grow locally on one lung, and one lung, the other lung, the contralateral lung, should be enough to live. So you generally, of most cancers, you generally die of the metastases, except for the liquid cancer like lymphoma and leukemias.
Okay, thank you for that. We have a question here. Are data from single subjects useful on their own, or must it be combined in cohorts to be predictive of outcomes?
That's a good point. So once you know once the classifier is calculated, they're sufficient and significant on their own in the context of a classifier. Otherwise, they're informative in terms of research properties, and sometimes it can provide insight. So for rare and infrequent disease, it may reduce the time to identify the key gene that's responsible if you have an assay that you think is the right assay. And we're doing that right now in coccidioidomycosis, the valley fever group led by John Galgiani. And that was our hypothesis when we obtained a grant 5 years ago, that there would be convergence at the pathway level between the Mendelian form of the disease, and there are just a few, there are 4 or 5 families known to have a Mendelian form of the disease, and the more common form of the disease, we're speaking of disseminated valley fever that is lethal unless it's treated for a lifetime. And the disseminated form, it's still an infrequent disease, but there's thousands of cases. So what we discovered, and the paper is in preparation right now, is that they share the same pathways as expected. The Mendelian disease were very interesting because it's like drawing a figure with a dot figure with numbers, right? And you draw your numbers and you could see the pathway. It's really illuminating these 5 Mendelian form that are on different genes. The genes are all first degree, their protein are all first degree interactions, stat 1, stat 4, in a known pathway of response to fungi. So this is very informative. In our assay, we did 2 assays ex vivo over the blood, and one of them was dectin that stimulates like the membrane of a fungus that stimulates a large number of the part of the pathway, and the other one was a lysate cocci that's destroyed, and the valley fever germ that's destroyed and put into the blood. And the response to that is convergent. We can say convergence between a Mendelian form of disease and those for which we don't know the inheritance, but it's not one of the known Mendelian disease, and we think it's a complex inheritance. So for us, it helps us deconvolute the signal to a much narrower number of candidates for which the effect size, if it's a complex disease, will be small. I know it's a bit complicated, but the genetics of that, and again, Mike Hammer is an utmost geneticist. He's identified new diseases through methods that were very difficult 8 years ago and are now more commoditized, I think, or there's more tools around it, let's put it this way. It still remains very difficult to know where to look because we all harbor a lot of mutations that may be unique, and which ones to look at that would be meaningful. And having an assay that we think is the right assay for the disease is very informative because, again, you can deconvolute the signal to look upstream at the DNA in only a few places. For example, for diabetes type 2, the studies are showing us that the polymorphisms that contribute to the familial inheritance of diabetes type 2, each contribution is about 2% of risk. And we know about 200, which cumulatively account for 16% of the risk. So it takes a lot of time to accrue signal when it's a complex inheritance, and it's better to know which genes you're looking at. Otherwise, it takes cohorts of 5,000 patients to 40,000 patients like we've seen in diabetes. And that's how difficult it is for the complex inheritance. We're still in the beginning of that.
Okay, we have a question here. The examples given are for prognosis. Would be great to hear about how methods may be used for analysis of treatment response.
Oh, I think I've destroyed the slide for this one. So we let me see if I can pull it up. Fine. I could pull up a different. So we've done a study on therapeutic response for HIV treatment, and that's the one I'm trying to pull up right now. I may have it in this one. So I'm switching presentation. Yes, and then I'll share the slide. Oops, this presentation here, and it's, sorry, I'm trying to get to this here. There you go. So this was a study that had very few patients. I think we had over a type of drug called the NNRTI, non-nucleoside reverse transcriptase inhibitor, that are supposed to elevate the CD4 T cells over a period of 3 months. The question was, could we, using the blood before treatment and 1 week after the beginning of the treatment, discover what's happening 3 months later, which is the elevation of the CD4. Since we didn't have enough patients to do a training set, it would have required at least 10 patients that do not fare well and 10 that fare well. That's 20. And we had, I think, a total of 16 patients here. And as you can see, it's not that they fare well or don't fare well. It's a progression, right? They fell. This one doesn't fare well at all. This one fares very well. And the other ones are somewhere in between. So instead, and we didn't have an independent set to validate it. Instead, what we decided to do is to touch the data only once. We would first verify if the transcriptome by themselves would evoke a response to CD4 using a very rudimentary tool called a first component analysis. It's from linear algebra. People learn that in first year of engineering or statistics or applied maths at university. And it's a very simple tool, and our goal was to take only the first component, which is the most important effect that's seen in the data in an unbiased way. It's not trying to train against the outcome, which is to the left here. It's only looking into the data, and it says, where is the data best organized to see the most difference between the patients? And this was the first component. So that's a sum. That was a sum here of 5,000 pathways, but we tried it first directly at the transcriptome, and the transcriptome would reveal nothing on the first, second, third. I think we went to fifth component and even combinations of components. But when we do the transformation of the single subject study, and we take the transcriptome of the two transcriptome of one person, we transform it into a pathway signal. So we go from 25,000 transcript, which themselves are organized in 5,000 pathways, and the signal is not to 5,000 pathways. You can see that it highlights a correlation, and the Spearman correlation is an unparametric one, so it's not based on a hypothesis of what's the distribution there, which is a very conservative way and correct way to analyze these data, and it's very significant. As you can see, there's a good correlation between the majority of the effect unveiled, unburied by single subject analyses, while it was buried in the transcriptome prior to the analysis. And that's a 2017 paper from Chika Lee and us. Thank you.
Okay.
So yes, yeah.
Somebody says, this is really fascinating. Do you have other examples of ways single subjects can serve as their own controls other than in cancer or infectious disease?
Yes, the single subject study and N-of-1 trials were conceived 30 or 40 years ago in Australia and in the US. I don't remember which name is from where, but one of the names from US, the other one's from Australia. I try to use both in my presentations. And they were done on usually one measurement. And what physicians would do is give the drug, remove the drug, give the drug, remove the drug, and they would check over time how that works. If you do it enough time, and there's not too much what's called carry-over effect of the drug in the period in which you analyze it, you can actually generate a statistic that that's based on what's called a time series. However, these are generally biased studies because it's hard to blind the patient of giving the drug or not, right? Unless you give them groups of pills that always look alike and they don't know which week they take what, and you're blinded yourself. But they didn't do that in the beginning, and they actually didn't use statistic. They used a visual method to look at the results. It was, I don't know how to say it politely, it was a relatively rudimentary method, let's put it this way. But since in the last 10 years, there's solid statisticians that have started looking into using time series in a proper way and designing the right trials. So that has evolved quite a bit, but it's based on 1 or 2 or 3 measurements or analytes. Generally, we call them analytes. they're working in very different ways. They need many more measurements over time for their statistics to work, but they measure very few things. The beauty of transcriptomes or metabolomes or methylomes, the dynamic part of the genome, or the products of the gene products. The metabolome is not actually a gene product, it's the product of the gene products. The beauty of that is that you have many measurements in one patient, and that looks a lot like a physician when they start trying to understand a patient. You have measurements that are incorrect, you have measurements that are missing, you have measurements that are correct, and you have a framework, a canvas in your head of what would be diabetes type 2. I can assure you there's not two identical presentation of diabetes type 2. Not at all. Some take their diet properly and their drugs properly and they don't respond, and they're twins with someone else that does everything wrong and they respond. There's even a case in COVID. There's a good case in COVID where the person that had the most severe disease was the one among the two twins, identical twins, living in the same city. I wonder if they don't even share the same apartment. So a lot of environmental factors are the same, and they were young, so there's not much change over time. And it's the twin that did not have hypertension and had a controlled diabetes that nearly died and required extra pulmonary oxygenation of her blood during a week till her lungs recuperated, while the other one had a milder disease and was out of the hospital in a week. And she had an uncontrolled diabetic, uncontrolled hypertension. So very, very confusing times right now with the interactions with COVID. And again, even having the same genetics, you can have a different response, and we don't know exactly all what's involved in terms of the environmental factors. Clearly, while we know that diabetes and hypertension increase your risk of the disease, what's been shown is that it's not necessarily whether it's controlled or not that increases your risk of the COVID disease. So I don't know if that answers your question, but clearly there's a lot of variation. There's a lot of things happening there. Mike, you're muted.
Sorry. Thanks. I'm sorry I was muted. Suggest ways we could use these methods to predict severity of COVID. And keep in mind, there are two aspects to what you're saying. One is the predictive aspect. You can predict a subgroup. Somebody belongs to the severe or the mild group. Then the second aspect, from all the way I see what you're saying, is that you can actually infer something about the pathophysiology by looking at the particular pathways that are differentiating the two groups. So how would you balance those two, and how would you suggest ways we could better understand COVID?
So conventionally, in statistics, we consider that there are two types of modeling you can do. You can do a classification, or you can do a group comparison. And you don't expect to find that the features or the molecules in our case here, or the pathways that are discovered in a classification would be meaningful to separate the groups. I know it looks a bit bizarre when I say that, but the problem is that there's so many measures with, let's say, 25,000 transcript or 5,000 pathways, that you may not have the driving genes or the driving pathways in your classifier. You may have the passenger one that's always correlated with the driver because they drive together in the same car, right? It's always, I have my spouse as a passenger, or I'm the passenger of my spouse, and we're always in the same car, but one is the driver, the other one is not the driver. And modifying the passenger will change nothing to the direction. Well, I mean, if we're not human beings, right? Robots will change nothing to the direction of the car. Clearly, if someone tempers with a spouse, I think we'd do something, but that's how they're conceived. So conventionally, that's a conventional wisdom. There's a lot of papers addressing the fact that people are confusing these two classes of methods for a long period of time after the genome was published, during 20 years, and they're still doing that. Here's the subtlety of the methods I've presented to you, and it's a really good question, because since we do single subject studies from the start, what I didn't tell you is a little trick we do. If a pathway is unaffected in all patients, we drop them from the analysis. And we're not cheating because we're not looking to the group of the patient. We're not looking to the class. It's just if a pathway is insignificant, let's say we're speaking of the asthmatic patients in response to rhinovirus. None of the asthmatic patients responded to, I know, 4,200 pathways. There were only 800 in which at least one responded. Then it's useless to use them in a classifier. That's called dimension reduction. And that's one of the legitimate way to do that. And it's very powerful. Additionally, I've told you, we can peek under the cover of an otherwise black box classifier because we did a single subject study in the beginning, and we know the significance in each patient and the effect size of each of the features, which generally is never provided by what's called a dark or black box classifier. But in our methods, we actually bundle the two aspects. We bundle the mechanistic aspect of, it's not really group comparison, but it's within subject comparison, right? Within subject, there are two conditions, and we bundle that with a classifier. So not to compare subjects across groups, but to compare a subject to itself, we can interpret that in that classifier, which is pretty unique. And that's why those were the first genome by environment classifiers in the literature, because they actually used two samples in one patient in two different environments. There's probably an application in which we can look across groups as well within the classifier, but we've not tried to unify the two techniques. What I've shown you in the presentation was just one slide of a paper that's in press right now, which was two group comparison. Remember when I've shown you the crosses that were great, not so good, but the single subject studies, the inter N of one was good. That's the slide of a paper in press where we've shown by simulation and retrospective study in two data sets and one prospective study that indeed we can reduce the number of samples to discover group comparisons to very few samples as long as we do single subject studies first. And the other paper I've shown you, which dates back from 2017, was machine learning over that as well. But there's a way eventually to bring convergence there. Well, classically, a classical statistician would tell you never to mix the two methods. We're doing that purposefully, but in an elegant way because they're separated from one another. And that's how we can do these, and we kind of balance the two.
All right, thank you. We're down to a couple more minutes, so I'm going to make a request that somehow we get the references available. I don't know exactly how we would do that. The presentation is being recorded and will be disseminated tomorrow for anyone who wants to go back and see the slides. Maybe someone will come up with an idea for the references.
The title in the presentation, under the title for every slide, there's the reference. Okay. I can show it. And it's not
that's good, Yves. We don't have time for you to go back. We could have time for one more question, but it's a head scratcher and you'd have to just sort of quickly answer it. We have just a couple of minutes left, but it says, I thought it was a very fascinating question. Do you have any idea about how this method could be employed for illnesses in which you can't access the tissue of interest, for example, in mental illnesses?
Okay, awesome question. A third of the known diseases are immune mediated. Even Alzheimer's is immune mediated, asthma is immune mediated. And by the way, what's going on in asthma, the immune cells never go back. So once the immune cells decide their fate, that's how we decide, and they go out of the blood vessels, like monocytes become macrophages. They never come back to the blood. And about 50% of the genes are expressed in all cells in the body, and they're subjected to local interactions that may make it a bit different, the response of the cell to the stimulus, but by far a great majority of the pathways seem to be altered similarly. So you lose about half the signal, but the blood is informative for probably all the cells in the body for 50% of the genes. But for immune-mediated diseases, and I'm not speaking of only infectious disease and autoimmune disease, I'm speaking of other type of diseases such as asthma and Alzheimer's. It's a good proxy, and you can use the peripheral blood as a proxy. I think the minimally invasive tissue we have are the skin, the blood, maybe local areas of entry of different orifice of the body, but not much more, right? So it's a very good question, but we think it's probably amenable. A third of the diseases are amenable to being approached by the blood, which is not bad. Knowing that there's only drugs for 2,400 diseases, and we know 65,000 diseases.
Yeah, I mean, I might just throw in one thing, and that's the blood-brain barrier is disrupted in many of these mental illness diseases and neurodegenerative diseases. So the blood might actually carry markers directly from the brain into the periphery. So I think
thank you all again.
Yes, thank you all for attending and asking great questions.
Awesome.
So, wow, lot of great information packed into an hour of time. Unfortunately, we're out of time for this evening, but we want to thank you all for attending, for your insightful questions, and again, thank Dr. Lussier and Dr. Hammer for leading us through this evening. Please join us for the final installment of this year's Precision Wellness in the Time of COVID-19 Symposium. That will be on June 21st, and it will be featuring Dr. John Ruiz. For more information on our Precision Wellness Initiative and our upcoming speaker, please visit the website address on the screen. Again, thank you for joining us and have a great evening.